all. This is Dr. Mubin Sayed from drbeen.com. Welcome to one more show. So the yesterday's talk about the studies in which we saw for long COVID, the interleukin 6, C reactive protein, interferon alpha, and then neurological um, blood biomarkers. There were two very common questions that I saw in the comments. One of them was, how do we treat this situation? And I've done a lot of talks about that. I think I'll do it once more. But the second one was, many people said, many cool beans said, my interleukin 6 has been elevated for some time, years, while I am a long COVID sufferer. And that also includes vaccine injury. And my CRP is normal. So that is what I want to address today, because some folks were concerned that this may not be a normal thing. So the short answer, in some people, CRP actually is normal and interleukin 6 is actually elevated. Normally, what happens is that once the interleukin 6 is released, it works on the hepatocytes or the liver cells, and the hepatocytes in turn releases C-reactive protein. So usually you can say that the interleukin 6 is responsible to help make CRP. So if interleukin 6 is more, then the CRP has to be more. But you would see in the study today that it is not necessary. So let's start our discussion. At the end of the discussion, I'll also discuss the role of CRP, not just as a biomarker for inflammation, but what does the CRP do? Why do we produce CRP? What does it do in our body? So we'll discuss that as well. So let's start. So these are the gifts of humanity. They're continuing. And if you wanted to participate in this all as well, this is drbeen.com with lots of cool videos and packages here. If you would like to buy the courses, we are almost done with the courses. We'll soon start selling these courses. It's actually me who had them not be ready in time because I had to write the descriptions for each course. And then at this time, while we are still working on the courses, if you would like to get access at a very, very reasonable price, look, $67 for access for all Dr. Been videos on drbeen.com is really cheap. However, this does not include CMEs. This is just access to the video content. Okay, so here is the study that we discussed yesterday, Biomarkers in Long COVID, a systemic review. Here is the study that I want to discuss today, discrepancy of C-reactive protein, procalcitonin, and interleukin 6 at hospitalization, infection in patients with normal C-reactive protein, procalcitonin, and high interleukin 6 values. Just the title itself tells you the situation. Then there are some more links in the description as well. So everything that you see here, including Dr. Bean's discounted price, these are the links present in the description of this video. So now let's start with my drawings. So yesterday, when we were discussing the C-reactive protein, what we were talking about, so I have to start from here. My apologies, I should have reset it in the beginning. So these are gifts of humanity, they're continuing. We were talking about the various labs yesterday. And then as we talked about the pathophysiology of the labs, we discussed that when we have the virus or the bacteria or just the spike protein itself in the vaccine, when they cause the trigger the macrophages or monocytes, then the macrophages or monocytes release a lot of cytokines or the chemical molecules. One of them is interleukin 6. And we discussed this, that interleukin 6 then in turn works on various tissues. One of them is the liver. Actually, these macrophages can actually be sitting inside the liver. And when the antigen is picked up by these macrophages, then they release interleukin 6 within the liver. And then that interleukin 6 acts on the liver cells within the liver, on the hepatocytes. And then in turn, hepatocytes release acute phase response or cause acute phase response releasing C-reactive protein. This is what we discussed yesterday. Today, I want to present to you a study. So here is the study. The study says that it was done on patients that were getting hospitalized by bacterial infections or fungal infections. And the study was done on the patients within the 24 hours of the hospitalization, and their C-reactive protein, procalcitonin, and interleukin 6 was measured. Of course, our interest today is the C-reactive protein and interleukin 6, so I'm just going to ignore procalcitonin. Then they also had another group of patients who had been in the hospital for 2 weeks, and they were also measuring their C-reactive protein, PCT, and interleukin 6. So these were the two groups. The question that was in the researcher's mind was, is it possible that whenever interleukin 6 is high, then the C-reactive protein will be high as well? Or there are other scenarios possible as well? So in technical terms, we can say that they were trying to figure out, is there a concordance only? Concordance will be that interleukin 6 is more and C-reactive protein is more. Or can there be discordance as well, where they are not dependent upon each other? The study includes patients from January 2015 to August 2021. And the numbers you can see here, those patients whose labs were done, and when I say labs in this discussion, these labs are CRP, PCT, IL-6. Of course, a lot more labs are done. So in this context of the lecture, labs simply mean these three. So 24 hours of admission, 7,149 patients, all these three labs and more were done. Then another 5,261, or even out of the same ones, 5,261 at 2 weeks of hospitalization, and their labs, including these markers, were done as well. What did they find? They found in 1.4% of the patients, 102 patients, who were in the discrepancy group, who were the group that were just landed in the hospital, they found, check this out, in 1.4% of the patient, normal CRP, normal PCT, but high or elevated interleukin 6. At the time of hospitalization, within the 24 hours, 1.4% of the patients had increased interleukin 6. And remember, these are the patients of bacteremia or bacterial infections, fungal infections. So they have inflammation going on. 1.4% have increased that interleukin 6, but their CRP and PCT are normal. So I'm going to leave you a thought over here. Why are they normal? It is possible that some people have their immune system built in a way that it doesn't respond with sufficient elevation of CRP. It is also possible that the interleukin 6 is released and it can be seen or it can be measured in the blood. However, the liver has not yet started producing enough response to produce enough CRP that can be measured above normal level. It could be unknown factors. And if you dig deeper into the CRP and interleukin 6 behaviors, you will find out that CRP is of two types. One, CRP that is released from the liver, which is a pro-inflammatory CRP. There is another type of CRP that is released at the tissue which is under attack or infection or damage, and that is an anti-inflammatory CRP. And I'm sure that when I say this, if this video goes up for CME, this would get canceled as well, because somebody is going to say this does not happen. However, these studies, you can simply Google anti-inflammatory CRP and you would find out that CRP molecule can be of two types, pro-inflammatory and anti-inflammatory. Normally, the molecule, when we talk about CRP, we are talking about the pro-inflammatory molecule that is released from the liver. So it is possible that the CRP is yet not released in the liver. And so that is why the CRP is still normal. And then it is also possible that over a longer period of time, not in the acute setting, but a longer period of time, the liver has decided or has become adapted to interleukin 6 levels and is not producing more CRP above and beyond the normal levels. So this is one observation they had. The second observation they had was the following. So of course, what they were trying to see was, is it possible that there can be low IL-6 and more CRP? Is it possible that there is more IL-6 and less CRP? So in the second observation, they had high normalized interleukin 6. And that interleukin 6 normalized, so it is normal, was greater than or equal to zero point 0.5. The number of patients was 349. And in these patients, the CRP and PCT were lower. Now, I want to add one more comment over here. If you talk with some medical professional or a PhD students or immunology student, they would say there is no concept of low CRP, and they will be right. CRP is usually not present if we do not have inflammation. So what we say is 0 to 0.8 is kind of normal. So something that is 0 is normal. What will be below normal? Negative? We can't have a negative molecule. So because of that, the idea of low CRP is actually not a valid idea. Still, here they say that CRP and PCT were lower. Now, CRP lower can be discussed in terms of previous level of CRP that was higher. And now that the inflammation is reducing, then the relative levels of CRP have become lower. That is possible. So here in the 24-hour group, 17.8% of the patients had lower CRP while the interleukin 6 were normal. 38.7 of the 2-week hospitalized patients had lower CRP and PCT while the interleukin 6 was normal. So this was another observation. Then they also saw in another set of patients who had normal CRP, and normal CRP 0.0 to 0.8, 0 to 0.8 milligram per liter, or actually lesser than even 10 milligram per liter is actually supposed to be kind of normal CRP. Between 3 to 10 is said to be normal but moving upwards. Lesser than 3 is supposed to be normal. It cannot be negative. So here, normal CRP, 24-hour group, 9.2% of the patients, 2-week group, 13.4% of the patient. However, these patients, this group, had in higher or elevated PCT and elevated interleukin 6. So the proof that this study provided was it is not necessary that there is always going to be a concordance when we observe or when we examine the interleukin 6 levels and CRP levels and procalcitonin levels. And it is entirely possible that there is more interleukin 6 present, which is an indicator of bacteremia, fungemia, or other inflammatory states. And CRP is also an indicator of a response to inflammation. However, they can be discordant, they can be asynchronized from each other. So this is the study. If I can very quickly flash that study for your viewing pleasure once more. This is the abstract that I presented, and then this is a very thorough, beautifully done study for your reading pleasure. The link is in the description, and please look at it. Now, shifting gears, another brief review of what CRP is or what does it do. CRP, just like I said that it can have a pro-inflammatory and anti-inflammatory molecular behavior, CRP is usually created from the liver in in a structure where there are 5 molecules of CRP that are bound together. This is called native CRP. 5 molecules. This is like, imagine a molecule is a ball. 5 balls clustered together are produced. That is called native CRP. When these 5 balls tumble around and they swim in the blood and serum and they reach the area of infection or area of inflammation, over there, these 5 balls dissociate from each other, they separate from each other, and they become monomers of CRP. This monomer of CRP will then start binding in the local tissue and help it participate in the inflammatory process. So what does it do over there? So let's see. One function of the CRP. So in these diagrams, the C here is the CRP. One function of the CRP is that it binds with the broken cells. It binds with them, which causes them to become opsonized. Opsonized means that a cell is tagged with some external molecules, normally antibodies or complement system. Here you are seeing a third molecule in a body that can opsonize, and opsonization will allow macrophages or dendritic cells to pick up this cell or pathogen and clear it out. Opsonization is kind of a signal for the immune cells to say, take this thing out. When I teach the medical students, I usually say that imagine we put a lot of candies on a pathogen, on a bacteria or a fungus or a broken cell. And the macrophages have a sweet tooth for this candy. They have a receptor for this candy, for opsonizing molecules. And they come over and they pick up that thing, bacteria, fungus, virus, broken cell debris, and they eat it up, they phagocytose it, and they digest it and they break it down. So CRP can opsonize the broken cells. It can opsonize the pathogens, for example, pneumocytes. Pneumocytes cause pneumonia. It can actually become coated on their capsule and opsonize them, which allows macrophages to pick them up and eat them up. So that is one function, opsonization. Then CRP can also cause complement activation. Complement is another set of proteins that are produced as the acute phase response or acute phase reactant protein set that is also produced by the liver, the complement. Complement's function, there are many complement proteins. What they do is they start aggregating on the surface of broken cells, on the surface of bacteria, fungi, and they make tiny holes in the bacteria or fungi, and they cause the holes then cause the substances to leak out of the pathogen or leak in the pathogen and destroy the pathogen. So here, if you see this little structure is a representation of the complement system, and CRP causes the complement activation as well. For the medical students, nursing students, NPs, PAs, doctorate students of, let's say, DNPs, what other structures help fixate complement? One very important structure helps fixate complement. Cool beans will know. So that will be IgG. Immunoglobulin G also performs complement activation. So anyways, CRP can also perform complement activation. So Parth over here said IgG. Absolutely, IgG does that. So here, the CRP is doing that. When the complement is fixated, that complement set of proteins will then gather on this surface, and they would make a hole, or some we call it membrane attack complex, or MAC. They will make a MAC. That MAC, or membrane attack complex, is nothing but a little pipe, microscopic pipe. That pipe gets inserted in the surface of this pathogen or the broken cell, and it just destroys that thing because it becomes leaky. Imagine somebody comes and breaks the walls of a house. So this is also something that CRP does. So CRP is not just a marker. Then, as you can see these beautiful diagrams over here, I am writing the immunology book, and these cells are going to be in the immunology book. So here, there are various immune cells. CRP can actually bind with various immune cells. For example, neutrophil here. How do I remember? I remember neutrophil, they have a burning furnace in them that does, that blasts the pathogen by doing oxygen bursts in there, in that, and when we'll talk about neutrophil, we'll discuss that. So here, CRP can become attached on the FC receptors. FC receptor or fraction constant receptors are actually receptors on the cells to bind with the antibodies. CRP can bind on the receptors for antibodies. And when it binds there, FC receptors on neutrophils or natural killer cells or dendritic cells or macrophages or B cells or T cells or whichever cells have the FC receptors, it binds with them and it activates them. Imagine the amount of activation of the immune system when CRP is attaching on the FC receptors. So that's also another function of CRP. The result of this is that these cells will become activated. And when these cells are activated, they're not only just performing their function, they're also releasing a lot of cytokines. Those cytokines will cause amplification of the immune inflammatory response, and the inflammation would just keep going rampant. Now, up to some extent, the CRP is protective. It is taking part in inflammation, which is necessary to handle the infecting agents. But when the CRP, for example, in SARS-CoV-2, when it goes through the roof, when it becomes too much, that CRP itself can drive the immune system to a crazy limit, which is not a medical term, to such an intensity that the tissue damage can occur. So CRP causes opsonization. It can does complement activation. It can induce inflammatory cytokines by binding to the immune response, immune cells. And then this is the diagram that was in the thumbnail of this video as well. That is the altered self. CRP can detect broken cells, deformed, denatured, altered cells. So I made this little cell. It doesn't really convey the message correctly. This cell is trying to do yoga and it is all doing splits and stuff, and it is altered. It is not what it was. So CRP can figure out, just like the pattern recognition receptors, PRRs or PAMPs, the pathogen associated molecular patterns, CRP can actually detect such abnormalities as well. And it can then bind with those cells. If there is a nucleus that is broken, for example, and then this little devil over here, the reactive oxygen species. So what happens is, imagine a tissue that is under attack and is inflamed or is ischemic, has blood clotting, and that tissue doesn't have enough oxygen and doesn't have enough nutrition, and the tissue is breaking down and the lactic acid is produced over there and the reactive oxygen species is produced, and the tissue is breaking and the cells are open and the proteins are, the guts are spilled outside. The reactive oxygen species would attack those broken pieces of the cell and deform them and denature them. And our own immune system would think that these are something bad. It will not recognize that as self. And the immune system would start attacking that, including the CRP binding with these altered self proteins, which in turn causes, as I said, it does opsonization, it does immune activation, so complement fixation. So when it would start binding with these altered self cells and the proteins and the nuclei, the immune attack would ensue, and we will have rampant inflammation if there is too much CRP. So these are the two parts of the discussion today. I hope it made sense. One was, can CRP be normal while interleukin 6 is more Yes. And second was, what does CRP do? And this is a completely amazing set of lectures that we can do. However, briefly, I explained that. So with this, thank you very much for joining in. Please like, subscribe, and share. In the description of this video, there are links. If you would like to get access to drbeen.com, there are links for that. You can buy me a coffee. You can use PayPal to support this work. If you would like to support this work, you can become part of Patreons or Substack as well. And talking about Patreon, you can even become a member of this YouTube channel as well. We will have an off tomorrow from the YouTube lecture because we'll have 2 Zoom sessions with the supporting beans, Substack, YouTube supporters, and Patreons, one at 1 p.m. tomorrow Pacific time, second at 6 p.m. tomorrow Pacific time. These will be the Zoom sessions. It allows me to have a little more heart-to-heart, one-on-one, and more open discussions in the Zoom sessions. These are not recorded Zoom sessions, so that we can just talk freely without any hesitation for what can be recorded and sent out. So if you like, join these places. All the links are in the description of this video. At least if you can like, subscribe, and share, that will work as well. I would see you on Thursday. Bye for now.